Welcome back to Scientia Academiae. Today, we're diving into the fascinating world of plant biology and dairy science. Buckle up as we explore how we can identify different tree leaves and uncover the secrets of cheese. The questions that we are going to discuss are from Paper 6, O-Levels Biology, June 2016 session. Question 3, 13 marks. Figure 3.1 shows leaves from 4 different trees: horse chestnut, laurel, hornbeam, and oak. In part A, let's take a closer look at the horse chestnut leaf and make a drawing. We have 5 leaflets. The lower 2 are smaller than the upper 3. The outlines feature clean, crisp lines, highlighting the serrated margins that give the leaflets texture. Notice the pointed tips, adding elegance and distinctiveness to each leaflet. Lastly, the realistic vein patterns branch out from the base, showcasing how the leaf functions in nature. In part B, we're comparing the shapes and margins of different leaves. For the laurel leaf, think of a smooth, elongated form, while the oak leaf presents a more irregular edge with distinct lobes. These characteristics help us identify each leaf type in nature. Moving on to C, Part 1, that is about the laurel leaf measurement. The actual width is 40 mm, and we need to measure it carefully in the drawing given. Draw a line to show from where the maximum width measurement was taken. The width measured is 1.8 cm, that is 18 mm. In Part 2 we need to calculate the magnification of the laurel leaf. Here's the formula. Magnification equals image size divided by object size. So, we calculate 18 mm divided by 40 mm is equal to 0.45. So the magnification is 0.45. Question 4, 11 marks. Now, let's shift gears to the topic of milk. Did you know milk contains reducing sugars? So how do we compare the sugar content in cows' and goats' milk? For the comparison, follow the following steps. 1. Prepare a water bath for safe heating. 2. Mix equal volumes of milk in Benedict's solution. 3. Heat both samples consistently for the same duration. 4. Observe the color changes after heating. The more intense the color, the higher the sugar content. Now, on to cheese making. When you add an enzyme and bacteria to milk, they work together to form solid curds. An investigation was carried out into the changes in pH during the formation of cheese. The pH was measured at the start and then every week for 6 weeks. The results of this investigation are shown in Figure 4.1. Part 1 states, join the plotted points on Figure 4.1 with ruled lines. As you can see, all centers of successive crosses are joined with ruled lines. Part 2 states, using the information in Figure 4.1, describe the changes in pH. As you can see from the trend, the pH decreases, becoming more acidic over time, with a rapid decline initially followed by a slower change. This trend suggests that while acidity increases quickly at first, the system gradually approaches equilibrium, as indicated by the data in Figure 4.1. Part 3 states, suggest an explanation for the changes you have described. Bacteria and enzymes work their magic to transform ingredients. Take lactic acid bacteria, for instance. They thrive on lactose, a sugar found in milk. Through fermentation, these tiny microorganisms convert lactose into lactic acid. This not only preserves the milk but also enhances its flavor and nutritional value. But that's not all. Other bacteria break down proteins and fats into amino acids and fatty acids. These transformations are essential in creating products like yogurt and cheese, showcasing the incredible benefits of bacterial fermentation. And that wraps up our journey from leaves to lactose. Don't forget to like, subscribe, and hit that notification bell for more insightful content from Scientia Academiae. Stay curious, and I'll see you in the next video.